Good morning, students. Welcome to our class, Biological Science. Dear students, in the previous class we have discussed about the plant tissues, animal tissues, cell structure and functions. We completed our 3 lessons. Now we are going into the new lesson. Before going into the lesson, I will recall the matter once we discussed in the previous lessons. Dear students, in this world, all organisms are made up of cells. Cell is the basic unit of life. Cell is the basic unit of life. All organisms are made up of cells. The cells have the ability to perform different types of functions like growth, reproduction, breathing, transportation, excretion. Okay, the cells can perform all the life process. We have already discussed in the first lesson cell structures and functions. In this lesson, we are going to discuss that how the cell is performing all these life process. The cell performs the all life process. To do this life process or to do these functions, it required various substances like the solids, like glucose, liquids like water, gases like oxygen. Yes or no? To perform the all life process, the cells require the solids, liquids and gases. So in this lesson, we are going to see that which materials should go into the cell or which are come out of the cell by the which part of the cell. Once you recall that one, which part or which cell organelle performing entering or exit of substances from the cell, that is none other than plasma membrane. Plasma membrane. So in this section, in this chapter, we are going to discuss about the functions of the plasma membrane. So the plasma membrane that present on the outer surface of the cell. This is the outermost layer that present on the cell. In plant cells, there is another layer that present around the cell, that is cell wall. But in animal cells, the cell wall, the plasma membrane or the cell membrane is the outermost membrane. So how we are going to discuss how this plasma membrane perform all these functions. Well, I am giving some examples: glucose, water, oxygen. Okay. Next, minerals, vitamins, carbon dioxide, waste. These materials go in or go out of the cell. Should go in or should come out of the cell. Glucose, go. Water should go into the cell. Oxygen should go into the cell. Minerals should go into the cell. Vitamins should go. Carbon dioxide should come out of the cell. Waste should come out of the cell. Okay? So different types of materials are absorbed by the plasma membrane to the cell. At the same time, these materials are passed out from the cell through the plasma membrane. So how this entering or exiting is controlled by this cell membrane. In this section, plasma membrane, we are going to discuss about the functioning of the plasma membrane. Before going to that, here is some of the experiments. I will explain about one is some experiments are here. To conduct these experiments, we need sugar solution. So, to make the sugar solution, we need sugar. Okay? So, add sugar to it, to the solution. This is sugar solution. Okay. So in the sugar solution, sugar is solute, water is solvent. We all know very well the water is the universal solvent. Okay, now in the sugar solution, sugar is solute, water is solvent. Already written there. Okay. Now we are preparing the saturated sugar solution. Saturated sugar solution. How do you prepare the saturated sugar solution? So take 100 ml of water in a beaker. 100 ml of water in a beaker. And add 1 teaspoon of sugar to it. Okay. Teaspoon of sugar to it and stir the solution till the sugar dissolves in the water. So we are making saturated sugar solution. Okay. To this add sugar to the water and stir it till the sugar is dissolved in the water. Okay. Do this one till a little amount of sugar is left. Little amount of sugar is left on the bottom of the beaker. On the bottom of the beaker. That is the saturated solution. You understand? Saturated sugar solution. So sugar solution which has satisfied and it can't no more dissolve the solute. No more dissolve of solute. Already it has dissolved some amount of solute in it. It won't have any ability to dissolve in the or to dissolve in it. You understand? Saturated sugar solution. So we add sugar to the water and stir it well until a little amount of sugar is left at the bottom of the sugar. This is saturated solution. Now we are going to concentrated sugar solution. Take 3 beakers with 100 ml of water. Okay, 100 ml. Add half teaspoon of sugar to 1 teaspoon of sugar to second, 1.5 teaspoon full of sugar to the third beaker, half teaspoon of sugar to the first beaker, 1 teaspoon full of sugar to the second beaker, and 1.5 teaspoon full of sugar to the third beaker. Okay. Among these 3, which one is more concentrated solution? The third one is the more concentrated solution. Which one is the less concentrated solution? The first one is the less concentrated solution. Can we make the concentration of the beaker 1 to beaker 3? Like beaker 3. Can we change the concentration of beaker 1 like the concentration of beaker 3? Yes. We can change it by adding another 1 teaspoon of sugar to it. Can we change third beaker, the concentration of third beaker, the sugar solution concentration of third beaker to first beaker? Yes, we can change it by adding another 200 ml of water to the beaker. The concentration of the solution is the amount of solute that dissolved in the solution or in the water. It is the concentration. Do you understand? The amount of solute that is dissolved in the solution is the concentration of that particular solution. What is the concentration of beaker 2? Concentration solution of the beaker to 1 teaspoon of sugar. That means the amount of solute that dissolved in the water or in the solution, that is the concentration of the solution. Do you understand? Now we are going to performing one activity here. To do this activity, we need beaker. So we need beaker. Take a beaker with water, okay, and keep the one dry grape or kismish. And keep one kismish in the beaker and leave it for about 2 or 3 hours or 4 hours. Do you understand? So our aim is to observe the different material. Aim in your textbook it is there, dear students, you can follow it. We can observe the different types of materials in the solution or in the water. Different types of materials in different solutions. So first take apparatus we required 2 beakers, dry grape or kishmish, okay, saturated sugar solution, like that. Next procedure: take the beaker and fill with water. Take one kishmish and keep it in the beaker and leave it for about 3 to 4 hours. After that, observe the kishmish. What has happened to the kishmish? Next, same thing. Take another beaker. Take sugar solution in the beaker. Here, water, normal water, tap water. Here, sugar solution. And keep a fresh kismis. Okay, and leave it for about 1 hour, 3 or 4 hours. What has happened? Dear students, in the first activity, if you observe carefully, the dry resin or the kishmish which is kept in the tap water, it bulges, it increases in its size. In the second activity, when we keep the fresh grape or fresh kishmish in a sugar solution, it shrinks. Kushinchukupoyindi. What is the reason behind that? In the first activity, the water moves into the grape. In the second activity, the water comes out of the grape. What is the reason behind it? Here, the layer that present in the dry kishmish or the fresh kishmish that makes the entry or exiting of substances that allow the movement of or exchange of materials. In the both sections, we noticed that the water is always moved towards the sugar solution. Sugar solution. The movement of water to the sugar solution. What has happened? From the less concentration to high concentration. So here, in the concentration, if you take the example of activity 1, the tap water contains less concentration, but grape contains high concentration. Because of that, the grape rises in its size, bulges in its size. The movement of materials, the movement of substances towards the sugar solution. Here, we kept a fresh grape, but the sugar solution, saturated sugar solution. What has happened? The water that present in the kishmish moves towards the sugar solution, saturated sugar solution. The kishmish shrinks in its level. This causes shrinking of kishmish. So dear students, we have to notice here or observe one thing. Water always moves towards the saturated solution, sugar solution. This means the movement of materials from one place to another place. The movement of solution from less concentration to high concentration. It is called osmosis. What is that? It is a Greek word. Osmo means pushing. It's a Greek word. Okay, the movement of materials from less concentration to high concentration. Okay. Okay. So, dear students, the movement of materials occurs through the plasma membrane. In the next class, I will explain another important experiment. Okay. Thank you.